All right, hope you had a good break. My name is Ryan Holloway. I work in Zahi Katz research group. I mainly work with different membrane bioreactor processes for potable and indirect potable reuse and for nutrient recovery. I've learned a lot the last couple days. For listening to you guys, I thought donor genes were the ones you got from Goodwill, and transfer genes were handed down from your older brother. Anyways, so I'm here to introduce Dr. Anna-Louise Reysenbach. Anna completed her BS in microbiology and botany at the University of Witwatersrand, South Africa, and received her PhD in microbiology from the University of Cape Town, South Africa. After which she did a portion of her postdoctoral work at University of Washington and completed her work with Normposium at Indiana, Illinois University. Anna is currently a professor at Portland State University, where she studies microbial life in the most extreme environments, including terrestrial and deep-sea geothermal vents. These deep-sea vents are located over a mile under the surface, and temperatures around the vents can exceed 400 degrees C. She recently returned from an extended research trip in the South Pacific using her deep-sea remote submarines, Jason and Alvin, and maybe she can explain why. She is interested in the biodiversity in these extreme ecosystems and how these ecosystems are tied to the geology and geochemistry of the environment. Her work into extreme environments has drawn interest from NASA, from which she has received grants and has served on the National Research Council's Space Studies Board. She has been a prolific publisher, being published in Nature and Science. I could not find the exact number of publications, but it's at least 10 pages on Google Scholar. She's an avid outdoorswoman, hiking the Oregon Mountains and sailboarding the Columbia River. And please help me welcome Dr.
Well, it's a great pleasure to be here in celebration of Norm's legacy, and I'm going to try not to cry tonight. So you just have to, okay, I'm going to change the subject maybe, right? That's how you do it. But you just have to go to a microbial ecology meeting to realize what a legacy he's left behind. It's incredible, and it's been wonderful to be part of his group. I can say it's hugely impacted my career. Being in his lab initially after coming from South Africa, he really showed me what it takes to be successful in science in the US. And there are just multiple dimensions in which I know I would not have been successful had I not participated in Norm's lab. Like Tom, I was so excited when that paper in 1985 came up with that lovely little drawing, that lovely diagram. And so I was very excited when Norm had a position advertised, which turns out to be Tom Schmidt's position. So I moved into Tom Schmidt's house. But I also remember when I first interviewed with Norm, and I was extremely intimidated. And we're at a bistro in Bloomington, Indiana, and he goes like this. So, what do you think about the origin of life? And okay, so very broad questions, and of course, ever since then, I've always thought I hear Norm's little voice always asking me these kind of big questions, and so it's just been a real honor. Thank you. And so what I'd like to do today is sort of give you a little flavor for how much he has impacted my research and my career. And hopefully you'll see the thread coming through what I have to say. And I'm told I'm only allowed 20 slides, which I don't think anybody stuck to that. So I'll try not to stick to it either. But anyways, so I came to Norm's lab very excited because I was trying to do this, and I knew when I looked under the microscope when I was working at deep sea vents, there are thousands of little blobs there, and I can only grow this one little weed called thermococcus. You know, it just was so frustrating, and so he really opened the world with his 16S ribosome RNA studies. My journey in his lab started at Octopus Spring, the famous Octopus Spring, and he's already mentioned the pink filaments, which I looked at them and I thought, wow, that's really cool. And they knew they had the 15S, but beyond that, we didn't know much. And so Jean Wicken, a graduate student in the lab, and I collected a sample, and we did the 16S ribosomal RNA flow library of this biomass. What did you say? Kilograms of biomass. But we just did a little bit. And sure enough, the clones fell into a lineage deep in the bacterial tree and were related to an organism that just had been isolated from a deep sea shallow marine vent off Iceland. And its name was Aquifex aeolicus. And everybody was very excited. New deep lineage in the bacterial tree. And voila, another one. The clones we called the electric monk clones. And thank you, Norm, for letting us acknowledge Douglas Adams' book that we were all reading at the time. And in that book, there's an electric monk, and it's pink. No, the electric monk malfunctions. And so it sees everything in a shade of pink and it believes everything. Read the book. And we all were reading it. Jim Brown had it and he gave it to us and we were all reading it. So these were the electric monk clones because they were pink filaments and we believed they were alive and good and living in hot springs in an octopus spring. So that was my first foray into the aquifers. While in Norm's lab as well, so there's the deep branch, he had this great instrument called the vent cap, which is not a very good photograph, but it's basically you knock down a hydrothermal chimney and you put the cap on top. It's a titanium cap, very difficult experiment, I must add. The first experiments had this huge, and I've got slides of this, but I'm going to show them, huge box on the side. We made it really side heavy, lopsided, and you could measure the temperature within this chamber, and the idea was to provide surfaces for these microbes to attach to and to grow under hydrothermal conditions. And so that and then also to look at succession. So first colonizers and then later how long they colonized. And so we had a few attempts at this. We then redesigned the thermocouple array and made this much smaller thing called the hobo probe, which is shown here. Yeah. Okay. So basically, you knock down a hydrothermal chimney, you put this vent cap on top. There's surfaces inside there that microbes can attach to. And well, we found one really successful, it's a very difficult experiment, one very successful deployment. When we opened it up, it was just goo, just microbial snot, would you like to call it, or goo? I don't remember. Goo, okay, microbial goo. And in Dan Frank's way, I was very good at mouth pipetting. So, a huge amount of biomass, and in that biomass we found a whole bunch of different organisms, but we also found a lineage that was related to some of the organisms who were working in Yellowstone, the aquifers. It was a new branch or new relationship with the aquifers. And so what was wonderful was that we were then able to grow this new aquifers from deep sea vents, and that then took my career along looking at the great diversity of aquifers throughout the world. And my lab was actually funded as part of that as a peat project. I don't know, we were the first bacterial peat ever to be funded at NSF. So that's another testament to Norm's ability to target good organisms. Since then, we've got lots of different ones. These are just a few, but their genomes have been sequenced from all over the world. So the beauty of some of these studies that we were able to use the search, so identifying microbes in the environment, and then actually targeting novel groups of organisms. And that's really what a part of my lab has really been focused on, is trying to put names and then actually trying to talk to Stan or Lola to figure out what these organisms are actually doing in the environment. We have continued with the vent cap type experiment. I'm not going to talk much about this, but I just wanted, I don't know if you've all seen these videos, but we changed the design dramatically. It's not a cap, it's now just a scaffold. And we let hydrothermal chimneys grow around these thermocouple arrays. And this is an example. We've knocked over a chimney, and within 2 days, we have a nice hydrothermal mush covered in microbial goo already, and we can then sample it. Very difficult to sample, but we've started to analyze the succession of microbes associated with hydrothermal chimneys. In some cases, the deployments have been longer, and I like to show this because it shows you how fast these hydrothermal chimneys grow. Some of them can grow 30 centimeters a day. And so it's a wonderful environment for microbes to quickly colonize and form really complex communities. So this one is a 10-day deployment, and the two little red dots are lasers that are 10 centimeters apart. So we knocked down a chimney, and this is what happened 10 days later. So you just keep going, and you keep going, and you keep going. So within 10 days, these hydrothermal chimneys grow to those that's 5 meters, and there's our instruments. So it was kind of a little bit of a difficult selection after that. Anyway, so we have been analyzing these kinds of samples, but what we're interested in is the microbial diversity associated with these hydrothermal chimneys at deep sea vents. And in the flavor of the search, what we're interested in is do some of the large-scale geological processes that we see at deep sea vents, and I'll explain what I mean by that. Does that change? Does that affect microbial diversity? Or does the environment just select? And why are we interested in that is that because up until very recently, we were only taking one chimney and analyzing the diversity associated with one chimney and one another chimney from another place. And we started to see just a pattern of who's there. A dominance of epsilon proteobacteria, methogens sometimes, mainly sulfur-reducing bacteria, etc. Okay, so we started to see some patterns, but it all looked like, yeah, we sort of get the same things in the same places because along mid-ocean ridges, the geochemistry is quite similar. Okay, so we were interested to see, is it really that similar? So we went to 3 different areas. The first is a typical spreading center, but it's a little atypical, and I'll explain it in a minute. The right, just very close to that hydrothermal system, another hydrothermal system called Rainbow is an ultramafic system, and that is where the deep mantle is pushed up into the Earth's crust and is much higher up. And so you get very different chemistries. We went to a backup basin. We've just come back from this area again. And this is where you get subducting plates. But at backup basins, it's very interesting too because in this particular area, you have typical deep sea spreading in the north and subduction of a plate in the south. So there's this very convoluted plate tectonics going on. The closer the subducting slab gets to something called the island arc, because you always get these nice volcanoes when you get subductions go along the west coast of the US. The closer you get to that subducting slab gets to the volcanic arc, the volcanoes start to influence the geochemistry in the hydrothermal fluids. Okay? So you've got a few different kind of geochemistries looking at these different sites. And then the third site was White's Basin, which is radically different. It's in the Sea of Cortez, where the continental sort of wash provides this huge sediment layer above the hydrothermal systems, and you get hydrothermally altered sediments, and so you get hydrocarbons so it's just cooked sediments that become hydrocarbons. And so when you go into this area, the chimneys are just oozing oil. The Alvin and the submersibles and the ROVs that come up are just dripping oil. It's quite disgusting, and it's quite smelly too. You can walk off with quite a bad headache when those systems come up.
Excuse me, Anna-Louise, yes. You're on a really interesting topic here, and this is high organic chemistry that's in water, deep in mud. The geologists tend to reckon that carbon comes from washing off either off the
Continental crust,
mainland
yeah.
or it's continental carbon. But I think it's possibly equally likely that it's microbial development in the sediments, which then gets cracked by the heat. We've talked about this before.
Yeah, but in
Do we know much
Guaymas,
about that?
I think in Guaymas, because it's Guaymas, which is right, it is so close to, I mean, it's in that basin, right? I think that's got a big, strong continental
It
influence.
certainly has that,
But it has everything else too.
There's a lot of petroleum in the sediments.
Well, the petroleum is because of the cooking of the sediments. Yeah.
Yeah, breaking carbon. And the question
Yeah.
really is what's carbon from? Is it the microbial development in the sediments or
Probably a bit of both.
alternatively washing?
Yeah.
Washing is usually the argument. But
Washing, but I think it's also, it's a high productive area, right? And so there's going to be a lot of ocean productivity in that area. So that's a marine snow coming on. Have I already taken 20 minutes?
No,
Somebody
no,
start.
you're not.
All right, so I just want to quickly give you a quick geology lesson. I'm not sure how many of you have. This is a very basic geology lesson, but think of ultramafic as situations like, as I mentioned, the deep mantle pushing up through the Earth's crust. So when hydrothermal fluid, okay, so this is how deep sea vents work. You get seawater that percolates through the Earth's crust, starts to react with the Earth's crust, and then gets with rocks. Stuff gets removed, stuff gets added, and then the hydrothermal fluid gets forced convectively back to the seafloor. Okay, so that's just the very basic. So if you're going through ultramafic rock, you're reacting with very different rocks, and then you get forced back convectively to the seafloor. So the hydrothermal fluids that come up are going to be high, very high in hydrogen. They have a fair bit of methane. They have little small organics, which is super interesting, and interesting from an origin of life perspective. I know something about the origin of life. Very high in metals, iron and copper, huge amounts, and very low pH. Basalt are typical mid-ocean ridges where you've got spreading, and you get the geochemistry is fairly similar everywhere. And then subduction is very interesting because as you have a subducting slab going down, it brings sediment with it, and so as that hydrothermal fluid circulates through that area, you're going to get fairly different type of chemistries. But you're also going to get, remember what I mentioned, when the subducting slab comes closest to that arc, the chemistry is going to be very, very different because the volcanoes are going to influence the geochemistry. So we were interested, okay, this spectrum, do you see anything in the microbial signature? Now remember, hydrothermal fluid is coming up, it's 400 degrees Celsius, there's 4 degrees Celsius water here, and you get seawater mixing with the hydrothermal fluid, and you get chimneys. So you've got a mixed water where the microbes are seen, this mixed water. So do you think you're going to see any difference? I mean, it's 400 degrees to 4 degrees, and that kind of range of chemistries, but by the time you mixed it, it's maybe quite similar, right? Well, the community is really different. Okay, so if you look at the hydrogen, just the hydrogen continuum from these different areas, so rainbow is that ultramafic site. The other end member from a hydrogen perspective in the hydrothermal fluid is Lucky Strike, also in this area. Guaymas is a sedimented region where there's a lot of hydrogen because of the cooking of the sediments. And then you've got this Backup basin that has everything that looks like regular spreading to this place called Mariner, which is my favorite place in the whole world. I forgot to tell you that. And it is the example where a hydrothermal system is most closely associated with the subducting slab and the volcano. Okay, so what did we see? So now we have the ability to not only do clone libraries, but we've got high throughput sequencing. So finally, we can actually really look in depth and at breadth at what kind of community structure do we see at all these different sites based on the hydrothermal chimneys we can collect. And we had to collect a lot of chimneys. It's not easy to collect a lot of chimneys to get statistical sampling to be able to do this kind of thing. But what we found, and I'm trying to hurry this along, what we found is this is a multi-dimensional plot, and I'm hoping we all, since we're all from the PACE lab, we all know a little bit about this, or most of us know about this, but each of these points represents basically the community structure, so a condensation of 50,000 or 5 to 50,000 16S sequences. And so the closer the 2 points are together, the more similar the communities of the samples we collected. Okay? And what was kind of cool that came out, and it was a surprise to me, is what we see is the sites where there's low hydrogen. Okay? So this is the Mariner area, which is it's low hydrogen because it's close to the subducting slab and the volcanic arc. Lucky Strike has very low hydrogen, if you remember that figure. Those communities are more similar, and the ones that are high hydrogen systems are more similar to each other as well. So we saw some separation, and then all the basaltic communities are fairly similar. Okay, so that was interesting to us. So what? Okay. Well, if you take a look at the actual details of some of those analyses, this is just an example of the Eastern Laos Spreading Center, which is where in the Western Pacific we've got the subducting slab. Mariner is this area down here where we've got this vent site is most closely related to the volcanic arc. And you can see that just from the colors, and this is just the arc here, I'm just showing that right now, but the diversity in these samples is very different from these up here, okay? And so that was interesting to us. But on top of it, there were some samples that gave us things we had never seen before at deep sea vents. This thing's called LART, because we actually cloned it. Amy Banter was in my lab at the time as my technician, and she cloned a sample that gave us a sequence called LART. And this has never been seen at deep sea vents. It's most closely related to Acrophilus, okay? From a Sulfatera. Okay? This is actually a different green, another organism never ever seen at deep sea vents. So we saw some different interesting organisms that we were curious about. In addition, I was very interested in the different vent arc here that we've been seeing from all different deep sea vent sites, and that seem to be endemic to deep sea vents. And one of these lineages was called the Deep Sea Hydrothermal Vent Euryarchaeodal Group 2. Okay, this later. Deep Sea Hydrothermal Vent Group. Okay, and this group we have found at every deep sea vent site that we've ever investigated, ever. Okay, it's always there. And nobody knew what it was doing. Everybody tried to grow it, I tried to grow it, nobody knew what it was doing. Okay, so, the LART, okay, so I'm interested in the DHVEs and the LART. And the LART appears on this beautiful sample now called the toilet bowl, because you can see it has a significance. There's the toilet. The toilet bowl this year is not as white. I think it's got a little grubby. Maybe we should analyze that. But anyway, it's a very interesting sample because the hydrothermal fluid is seeping through this really spongy rock. It's about 110 degrees Celsius up in the top here. The geochemistry is superbly unusual because it's a pH of 2.5, very different from any other deep sea vent, and it looks like pure hydrothermal fluid. So it's got we look for magnesium in the hydrothermal fluid to see how much mixing of seawater has happened. There is no magnesium in the fluid there. So what has happened is that fluid is true, deep hydrothermal fluid that has cooled, but not mixed with seawater. So it's pure hydrothermal fluid that is cooled. In the process of cooling, minerals like iron have precipitated out. And so it's the most similar, deep, subsurface, high temperature fluid that you could ever imagine to sample at 110 degrees. All right, so we were interested in this larch, which also grows on this sample. So we said, well, could we grow this picrophilus type organism? And so I always show the slide, but I don't think you've seen this slide, Norm. This is an example of how difficult it is to grow things that you really want to grow, okay? So this was some Aquifoliaceae we were trying to grow in Yellowstone. I had all this different media, and I inoculated all these tubes, and then put the rack in the hot spring in Yellowstone because that's the right incubation temperature, and I came back a few days later, and more things had grown on the outside than on the inside. Okay, so the question is then, do you think we grew the lot? No, not yet, and I love to tell the story. But we have somebody even more interesting. Well, we still have. And we grew the devil heterotroph vent blob with 2 horns. So this was so exciting because actually Amy Banter was in my lab at the time. She had been for 4 years analyzing the DHVs from all these deep sea beds. We had this great way to say yes, they're here, and no, they're not here. We had really great data, and we tried to grow larch. And I was at NSF at some panel, and she calls me up and she says, Anna-Louise, do you know what Mr. Lou got? It's not larch, but it's the DHVE. And it was exciting. It was, so we got something that we weren't really looking for, but we got it. Anyway, so the DHVE now are easy to culture, very easy to culture actually, it turns out. And we've got numerous genomes of these, and we continue to grow these from different places around the world. They do show a strong biogeography as well. And we still don't have the larch. We're trying again. We've just come back from a research cruise, got really beautiful samples, and multiple labs are now trying to get the larch. But in the process, we've gotten some other interesting organisms. For example, the most moderate thermophilic photogenics. Again, genome's been sequenced, so this adds to the big database that's being generated. All right, so I wanted to move just full circle because this shows, like Amy, our paths sort of tend away from Norm's lab, and then they always come back. And curiously, in all these places that we visit, we see these things called a core archaeota. And Sue Barnes is up there, and I was in the lab when that first broke. We also see a lot of these very cool little organisms called nano-archaeota. And so, coming full circle, we were funded fairly recently to start expanding the diversity of the nano-archaeota. And I'll explain about the nano-archaeota. But it was the second microbial type, well, this time the first archaeal peat grants awarded by an NSF. Peat is partnerships, experience, blah, 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 taxonomy. And enhancing experience in taxonomy. And it just, we've got all this microbial diversity, and a lot of the proposals that are getting funded or have been funded in the past at NSF still notoriously, or not notoriously, but historically have not been microbial. And so this was a really great opportunity for us to explore this interesting group of organisms. Okay, so the nano-archaeota, I think some of you are probably aware of it. They were first described in 2002. Many of us who have looked at some of these samples under the microscope always see these little tiny things that light up under your microscope, tiny. I used to call them the pinprick cells as I was trying to grow something, and then a few transfers, they'd be gone. Gone. So, I don't know what they were. Anyway, Karlstetter knew about them, and he very carefully and very methodically was able to isolate this group of organisms. And it turns out that they form either a symbiotic parasitic relationship with another archaeum, in this case, Ethnococcus hospitalis, a autotroph. When their genomes were sequenced, they turned out to be very interesting. They have split transfer RNAs, which got Dieter Sol very excited about. They didn't have, the nanoarchaeum didn't have an RNase P. And the idea that they had split genes was thought to be an ancestral character, and their phylogeny is very unsettled because are they derived or are they really ancestral? And so we've gone ahead and analyzed these things, and this is a collaboration with Mircea Kodar at Oak Ridge National Labs. We've gone and analyzed, and this is an old slide, but it's just a preliminary slide just to show you that they're all over the world in terrestrial and marine systems. And so we wanted to get a new one, and so we first went to Yellowstone National Park, and we went to the famous octopus, this is now the obsidian pool. And this is where Mircea was able to get an enrichment of a nanoarchaeum, and then used single-cell genomics to obtain a new nanoarchaea from octopus spring. And when the genome sequence showed that they were fairly closely related to nanoarchaeum equitans, but they had a very different host, okay? Very different host in the sulfolobales. The single cells were almost completely sequenced, and now Mircea just recently got a pure culture of this, and we're preparing the manuscript on this, and the genome is complete. So just to show you a phylogenetic tree, but you can see that they have two different hosts in this case. Of interest from the genome sequence.
Previous slide.
Uh-oh.
Well, no, it's interesting. Chuck Robertson has done a lot of treating with nanoarchaea as well. And we find that it's clearly urine. The notion that it's some sort of a deep divergence. We just don't see that at all. And I see that you're also.
Yeah, oh yeah, it's, yeah, I totally don't believe it's, I think it's derived.
Yeah, well, I also think,
yeah.
We're in agreement.
Yeah, oh yeah, totally. And we've actually got more data now, and even more, which is what I'm going to end my talk with. But so actually, so this is the single cell tree, and I'll show you shortly another tree which has not only the single cell tree, but then the culture from Yellowstone, which the culture turns out it was from a different hot spring in Yellowstone, has a different host, and this is slightly different nano. So the good news is we're able to grow these little elusive nanoarchaeas, but they're they hurt my heart a lot of the time because we do lose them, and I can talk about that at length. It's the reason I've got a few more grey hairs now. Anyway, but what's interesting from the comparison of the genomes is that the new Yellowstone nanoarchaeotes are a little different. Okay, they don't have the trans kind of split transfer RNAs that got Dieter Sol so excited about with the nanoarchaeum equitans. And the other interesting thing is they can do full gluconeogenesis. We were able to identify an RNase P. I don't know what that is. That's, I mean, maybe there's an RNase P in equitans that we can't, the motif we don't know of. This is interesting and curious, and I'd like some comments about that. But both the closed genome and this draft genome, there's no ATPase synthase that we can detect. Okay, so we have a new one from Yellowstone, very exciting. How about down in New Zealand? New Zealand would be a nice alternative. So we went down to New Zealand, sampled some hot springs there. There's Mircea Kodar, my collaborator on this. And this is at Tikiteri. I don't know if any of you been to hot springs in New Zealand, but this is kind of a mud pots area. And we got several stable enrichments, and this is just a sort of a diagrammatic representation of some of the clothes and then some of the enrichments we've got. And here is the Yellowstone ones. This stable enrichment it's an enrichment, and we look at it, we get like all excited about it because we've got nanos in here, can't see them, we can't fish them. We also have these nice, thin, massively long rods, coxi, and then regular little plump rods. And when we sequence the enrichment, we find the Chorochiota, we find Pyrobaculum, it's like a swear word to me in a way, an organism called Phavidococcus. So mainly 3 archaea. So one of these, and we get nanoarchaea. Okay? So one of these is the host, or maybe all of them, of the New Zealand nanoarchaeota. And so this is just the tree. You can see here's the New Zealand nanoarchaeotes. We get Phavidococcus, Pyrobaculum, and the Chorochiota. And this also just shows the other Yellowstone ones. We have not been able to yet identify the host of the New Zealand nanoarchaeota. As of yesterday, I think I do. But I'm not going to tell you because I'm going to jinx it. But I got some sequence back yesterday. We actually collected New Zealand samples, and I did a bunch of sequencing and enrichment cultures, and what we did was we got rid of anything. No, I'm going to tell you this. Alright, but this is the old culture, the old enrichment. So there are these lovely long thin rods, these big fat things called pyrobaculum, and then these things which Amy Buck probably says are vesicles. Okay, I suspect they are not, but it's possible because there are some where you can see some, vesicles that are clumped. You can see vesicles, nanoarchaeotes that are assembled in these ways, but we don't know that yet. And until we can do fish, and until we can really show that these are the nanoarchaeotes in these samples, that'll be very difficult to not say that they weren't vesicles. But what we did was we've gone ahead and done metagenomes of these enrichments and pulled out, and this is again very preliminary, this week's data, we pulled out the nanoarchaeotes and just compared them. Now, again, this is we did this at sea, so because of we can't work online at sea, we were limited to memory on our own computers, so we just limited it to context that were 2,500 base pairs. And so this will get refined. Okay, so we know this is the beginning of a metagenome study, but what is kind of cool is that we've seen most of the players. We've seen very similar transfer RNAs. Again, it's draft, as you can't totally say that something's missing. It's not there. But then when we look at some of the features that we saw in the Yellowstone nanoarchaeotes, you can see that there's some similarities. And in particular, we detect an RNSP in our New Zealand strain. We also cannot detect an ATP synthase in our New Zealand strain. Now, as I said, it's not a complete genome sequence. We do not see any split transfer RNAs, and we see flagella genes. So interesting. There seems to be some similarities between the terrestrial and the nanoarchaeotes and the marine differences with the marine ones. And in addition, we went ahead and tried to see what are the cores doing. Okay, and so the cores are not very exciting. Sorry, as yet. We got, again, it's a very preliminary study, but we have some primary data. And then if we look at what is absent in cryptophyllin, it's also absent basically in our draft genome, so maybe it could be there, but it's possible that these are pretty similar. The 16S's are very similar, the New Zealand one versus the Yellowstone one. So this is very interesting. I then got very excited because I thought, okay, that lovely thin filament is the core archaeote, and I'm surely I can isolate it. And you can see I've got this beautiful, not very beautiful, but a TM, and lots of very thin little rods in a almost pure culture. Well, I can tell you that this organism is not the core archaeote. And so it's a very different thing and a very interesting thing. You look at your culture and it's totally clear because they're so thin they don't refract the light. And so, but it's a crowded culture of these very thin filaments which are known as thermophyllin. So, oh dear. But we will get the core archaeotes, but I think the core archaeotes we're going to have to just keep in their enrichment culture because it looks like they lack some of the key enzymes for making certain cofactors, for example, and so somehow we're going to have to work on that. So, what I just hope I've shown is that there are many wow factors, because I think the DHV are wow and the nanoarchaeotes are wow, waiting to be discovered through the search. And I also want to thank very, very much Norm. And I looked for a picture of him and I couldn't find one. And so I used the more professional one because the one I had was the frontal view. He probably doesn't remember that one. He was like this. And, anyway, but it was really fun working in your lab. I continue to love working along these lines, and I know all my enjoyment for doing the research I do was stimulated very heavily while I was in your lab. So thank you very, very much. And for those of you who didn't know, there is now a PACE, and it didn't translate on you, but that's okay, because I don't know if I want to translate it, but it says PACE archaeota, WOES archaeota. I'm not sure you all know about the PACE archaeota and the WOES archaeota, but there's a new paper out that describes these new groups that I'm not sure we all agree on. So with that, thank you very much, and thank you for inviting me. Again, it's been an incredible honor to be part of the family, the Greater Place Lab family. My work is funded by the National Science Foundation. The work I showed you was done primarily, my culturing guru is Mr. Liu, who is gone into retirement, come back out of retirement, gone into retirement, come back out of retirement, and he's still working for me. Norm, you can come work for me. He's the same age as you.
Good idea.
And John Kelly's been doing some of the metagenomics work. Gilbert Flores was a grad student in my lab that did a lot of the diversity studies. These are collaborators that we're working on some of the enrichments. And then, of course, the people down at New Zealand have been incredibly good collaborators to try and nail this little New Zealand guy. And then Mircea Pradar is my main collaborator on this, but the people in his lab are also very valuable in some of these studies. And thank you, and I'll take any questions. Yes.
So this is the classic picture that we think of when we think of nanoarchaeota. But in your electron micrographs, all of the nanos were off of all the other cells.
But I got some others that they are on there.
But my question was going to be, couldn't it be simply a free-living nano?
Oh, you saw I put a question mark.
Yeah.
Yeah.
Does it have to
Okay,
be
so a lot of the electron microscopy that we did was opportunistic. So I'm a micro too, I'm opportunistic sometimes. And so those were, I would wait, because because the nanos get to high abundance late in the exponential phase of ethnococcus. And so I really wanted to make sure there was enough biomass, and it was just a negative stain, so it's very easy to do and quick. So those were very encouraging for us. But what we really want to do is get our qPCR assay to work well. That's a sad story. To anybody who wants to hear about it. Months of work with, you know. Anyway, so that's just promising data. But we have them associated with the pyrobacterium cells. But that's, I don't know. I do not know if they're vesicles. Or viruses, they're 100 nanometers. They're 100 nanometers. The nanoarchaeotes from Yellowstone have associated viruses with them, is what I hear. So it's possible, which is maybe why I lose them. But it's a really challenging project, but it's really fun. Any more questions?
Now about this origin of life.
That's how he said it. I couldn't do it, you know. Now about this origin of life. What do you think about it? Thank you.
Thank you, Anna-Louise.
Okay.